keynote speaker, Dr. Brindha Devi. She is Associate Professor at Queen Mary's College, Chennai, and she has over 13 years of experience in fishery biology and biotechnology. She applies modern biotechnical techniques to promote sustainability in the fishery biology, and her qualifications are MSc, MPhil, B.Ed, PhD, and so on. And she has an advanced research and interdisciplinary studies in zoology. And Dr. Brindha Devi is our long-term association with ISGCT. Can I invite? Oh, madam, you're here. Yes.
Thank you, Bona. Good morning. I'm a teacher. Good morning, students. That's it. Okay. Let me start like this. Kripagaran sir was asking you, are you all biologians? I could see only a few hands raising, right? Now I'm going to call you. Are you all biryani eaters? See? Such a josh, like what Lalit ji sir said yesterday. I'm seriously asking you, are you all biryani eaters? Yes. Do you know what is seasoned there in biryani? Your mom does, isn't it? When seasoning, she puts oil in the frying pan and or the cooker, and she puts something inside that. So I'm sorry. What's it? Can you? Anyone? That's my research topic here, and I've done some molecular docking with that biryani material, something inside that biryani, and you'll see next. So my topic is molecular docking, and as the engineering students, you might be knowing what is docking. That is, it's a binding interaction between the target and the ligand, which makes a complex. It might be a protein, or it might be a nucleic acid or ligand. And this is particularly used for drug development that is in pharmaceuticals. And the hero of the research is going to come in the next slide, please. Can you see what it is? Yes? Have you all seen it? Yes, it is star anise. You put a masala or some spicy substance inside the biryani material, isn't it? So this is star anise, and we took it for our research actually. The scientific name of star anise is Elysium verum, and I did some docking studies with this Elysium verum. It is a very, very, very flavorable substance, and essential oil is being prepared out of it, and they have predicted some structural conformation. That is the next part of this slide. Next, please. And have you all known that there is a very interesting material in this star anise, that is, it is called as alpha bergo. What is it? Alpha bergamotine. It's an anti-cancer agent, and it is not only an anti-cancer agent, it is also a highly, it is an antibiotic against the pathogenic organism, and here our pathogenic organism is Aeromonas hydrophila. And we did some studies, biochemical studies, and we found out that there are certain compounds, out of which there is one compound, I've highlighted it here. It is sesquit. Can I have the hand mic? It is sesquiterpenoids, and it has alpha bergamotine. And we did some docking studies with that, and we found that that it is against some pathogens, as well as it has that is, it protects us from the cancer. And we interacted this alpha bergamotine and some that is the medicine which we use, like doxycycline as well as tamoxifen, which goes against the breast cancer. And we did this docking studies. Next, please. And our docking studies are here. And this is a part of my research work with my PhD student, Dr. Mogna Sundari, who's colleague of those invitees here. She's my student. Yes, next slide, please. And we did some in silico docking process here, and the data was retrieved using the SwissProt database, which is relevant to both Aeromonas hydrophila as well as the breast cancer protein. And the structure was predicted using the Swiss model. 2D as well as 3D structures were predicted. Next slide. Now, For the ligand preparation, we took two ligand molecules. One is tamoxifen. That's a small selective estrogen receptor modulator, which is used to prevent the breast cancer, which is a medicine which is used for breast cancer. And then the doxycycline, as you all know, you take it for, it's an antibiotic. It is against the pathogen, it is used for the pathogenic studies. And we retrieved 2D structure as well as we drew some 3D structure using the ChemDraw. And the protein ligand docking was performed using the autodock method. And the autodock method was, it was a semi-flexible docking approach where we retained the protein as rigids and then the ligands flexible. Next slide, please. Now, you could see the, that is, I told you we drew the 3D structure using the ChemDraw. And the 3D structure of doxycycline is here, tamoxifen, as well as alpha bergamottin is here. And it's there, see, it's you can see the 3D structure and the functional groups of these all 3D structures like doxycycline, tamoxifen, as well as alpha bergamottin. Next slide, please. Coming to the main area of the research, we did the protein ligand mechanism of these, that is the protein which was chosen by us, and as well as those two ligand complexes using the autodock 4.2, and the allowed degrees of freedom for the ligand molecules. I told you the ligands are flexible and the protein is kept rigid. So only the side chains can move, side chain residues can move in the protein, otherwise the ligands are very flexible and the degrees of freedom are 10. And we used two types, that is box type as well as grid box parameters. And to predict the active site was also depicted out of it. And we made an exhaustiveness of 100 so that we could get the best output out of it. Next slide, please. Okay. Now, there are two more interesting things which was identified by us. Like, see, actually, we made an interaction of this tamoxifen as well as the doxycycline with two more proteins. That is, there was a transport protein and structure. I did not mention it here. That is the second point. Transport protein as well as a structural protein, which was used to combat the Aeromonas infections with doxycycline. And the docking result was also compared, the binding modes were analyzed with caspase 3 as well as BCL. BCL is breast cancer lymphoma 2 proteins. And we found out the potential synergistic effects of these proteins using the docking studies. And you can see the ribbon-like structure of these two proteins, that is the structural protein and the transport protein of Aeromonas hydrophila, which is a pathogenic organism. So you could see that the transport proteins in Aeromonas hydrophila, it facilitates the movement of the molecules like ions, atoms, everything across the cell membrane. And from the protein data bank, we retrieved this model. And then we also had the secretion protein. That is, it is type 3 secretion system. You call it as T3SS complex from the Aeromonas hydrophila. It is a structural protein. It also enables, that is, it injects the, with the help of only this T3SSS, Aeromonas hydrophila, it injects proteins directly into the host cells and establishes infections. Next slide, please. And, okay, there comes the docking studies. And we performed some docking studies using PyMol. And the docking scores we got was this one, that is minus 6. Minus 6.2 for alga bergamottin, which is docked against transport protein, and the structure is there. You could see the side chains with the amino acid residues. I'll explain it to you next. The next slide, please. Docking score of alpha bergamottin against the structural protein of Aeromonas hydrophila, it is 4.8, and you could see the docking between the alpha bergamottin as well as the structural protein. Next slide, please. And the next is the docking between the doxycycline against the transport protein, that is 6.9, and you could see the docking structure there. Yes, next. I'll quickly just move on. the docking score between the doxycycline and the structural protein. I've been telling about the transport protein and the structural protein, and we have been doing docking against alpha-bergamottin as well as structural as well as transport proteins. And we are also comparatively doing studies against doxycycline with structural protein as well as the transport proteins. Next. Now, we made the docking processes. I'll explain later to what happened about the docking processes. And next study of us is we did docking against caspase 3 and the BCL2 protein with the MCF7 cells. And caspase 3, you can also call that as Cas329306. It is a group of enzymes which plays a role in apoptosis. And it has a name in protein data bank, that is 2XYG. It is a unique identifier. And as well, there is B-cell lymphoma 2. It is, 2 is also a protein that plays a very good role in apoptosis. And you know, apoptosis, it is a programmed cell death. Next, please. And now, like doxycycline, which is compared to alpha-bergamottin, we are also comparing alpha-bergamottin with caspase 3 protein as well as against caspase 3 protein as well as BCL2 protein, and these are the docking scores. And you could just see the docking score, that is 6.6, and how it is docked here. Next. The docking score and edge bond interaction of alpha-bergamottin with BCL2 protein is here, and it is 4.43. Yes. Then I told you about the ligand, that is we are using against the protein. So that is tamoxifen against the caspase 3 protein. It is 6.98, the docking score. The next one is tamoxifen against the breast cancer lymphoma protein 2 protein. The docking score is 6.5. I hope you will remember all the docking scores now. Then we'll be going for the explanation. Next, please. Now, so comparing the docking score between the alpha-bergamottin and the doxycycline with aromonas, we are going for only infectious proteins. And alpha-bergamottin against the transport protein, we saw the docking score was 6.2 kilocalories per mole. And you could have seen those hydrogen bonds there. I told you hydrogen bond interaction. So 3 hydrogen bonds were there with specific amino acids. So the amino acids are named there: tyrosine, isoleucine, tyrosine as well as valine. And then where alpha-bergamottin was also docked against the structural protein, you could have seen the docking score of 4.8 kilocalories per mole. And 1 hydrogen bond was formed with the specific amino acids, that is tyrosine, arginine and valine. And doxycycline was also docked against the transport protein, and the docking score was 6.9, and 2 hydrogen bonds were formed. Then the doxycycline was also docked against the structural protein, and docking score was 7.28, 2 hydrogen bonds formed with specific amino acids. So that was for the pathogenic studies, and we did a separate study with tamoxifen for the breast cancer cells, and MCF7 was our breast cancer cell that we took for our study. And we docked it, and alpha-bergamottin against the caspase 3 protein, the docking score was 6.6 kilocalories, and it formed 3 hydrogen bonds. You should note that the hydrogen bond interaction is very, very important in predicting the docking scores. So 3 hydrogen bonds formed with specific amino acids. Then the alpha-bergamottin also against BCL2 protein, the docking score was 4.43 kilocalories per mole. 2 hydrogen bonds were formed. And tamoxifen against the caspase 3 protein, as well as tamoxifen against the BCL2 protein, you could see the docking score as well as the amino acids involved in it. Next. So we made a comparative docking analysis of this alpha-bergamottin, tamoxifen as well as the doxycycline. And the alpha-bergamottin exhibited superior docking potential among the compounds which are tested. So it had the highest docking score of 6.12 kilocalories per mole against the transport protein as well as 4.8 kilocalories per mole against the structural protein. And as I told you, hydrogen bond interaction is very, very important in this docking process. So alpha-bergamottin forms 3 hydrogen bonds, whereas tamoxifen as well as doxycycline demonstrated only a few hydrogen bonds. So tamoxifen formed only 1 hydrogen bond with the caspase 3 protein and 2 hydrogen bonds with BCL2 protein. whereas doxycycline formed 2 hydrogen bonds with both the target proteins. So variations in this docking potential as well as the hydrogen bond formation was attributed to the chemical structures as well as we did the functional groups, and we also found out the molecular properties of these compounds. And these docking results are we also maintain that is specific amino acids were also, that is I told you about the amino acids were within the side chain residues. It was also involved in the hydrogen bond formation, and it contributed to the binding efficacy, and it maintained the stability of the ligand protein complexes. So these docking results which we performed was insightful on computational simulations, and it also formed potential interactions. Next slide, please. So we made some GCMS characterization of the methanol extract of Elysium verum, wherein the alpha bergamottum is understood to be therapeutic. It had therapeutic properties, and it was used for docking studies. And the protein ligand interaction and the docking scores were evaluated. And from this computational docking studies, we ascertained the antibacterial as well as the anti-cancer properties of these this particular compound alpha bergamottin, which was characterized by GCMS in the extracts of the Elysium verum. Next. So I think you didn't see this. So I asked you in the first, before asking my doing my presentation, are you all biryani eaters? Yes? Yes. So you will never forget this. What compound? Alpha bergamottin. Alpha bergamottin, which is there is in your biryani, right? It is an anti-cancer agent, and as well as it is a agent against the, it has an antibiotic property. It is an agent against the pathogenic organism Aeromonas hydrophila. So put your knowledge into this, and you can also do more research on this, right? Thank you. And next slide. There is a paper, this is a prestigious paper we published in Fish and Shellfish in Immunology, me and my student. It's an Elsevier journal, and we made all the studies. Apart from this docking studies, other studies were also done in this. Thank you.